So the first molecule we use in DNA replication is DNA gyrase. So what that does is it uncoils the DNA so that helicase can come in and break the hydrogen bonds between the bases. So helicase moves down like this and it opens the DNA strands. So what helicase does is it creates a replication fork and it splits the two strands apart, and to keep the parent strands separated because they want to attach back together, you have to attach single-stranded binding proteins to the outside of the parent strands, and this just makes sure that the strands don't come back together. So the next molecule that comes through is DNA primase, and what that does is it adds a primer made of RNA so that when polymerase 3 comes through, it knows where to start putting DNA nucleotides. So starting with the leading strand, the DNA primer comes through and adds the RNA primer. So it comes through, and with the leading strand, it starts on the first nucleotide and it adds that primer so that when DNA polymerase comes through and it comes down the DNA and reads this primer, it knows that it needs to start building DNA. So it adds the nucleotides and it keeps going in that direction towards the replication fork. And that's just for the leading strand. So it's important to know that for the leading strand, when the DNA polymerase 3 is coming through and using the parent strand as a template and adding the daughter strand, it moves in the 5-3 direction of the daughter strand to add the nucleotides. On the lagging strand, everything's a little different. So when the primase comes through on the lagging strand, it adds different pieces of RNA primase throughout the DNA. So it comes through and it adds these different primers throughout the DNA, and the DNA primase is working simultaneously with the DNA polymerase 3. So what that does is it comes through, and when it reads the primer, it knows that it needs to start building the nucleotides. So when it reads this primer, it starts building them like so and matches the bases, and then when it hits the other primer, it knows that it needs to jump back and find another primer and keep building the DNA. So it keeps doing this, and then again, when it hits the primer, it knows that it needs to jump back and build more DNA, and it keeps doing this until the strand's complete. So once the parent strand is completely duplicated and it's done, then we need an enzyme called DNA polymerase 1. So what that does is on the lagging strand, it goes through and goes in the 5' to 3' direction, and as it moves along, it replaces the RNA primer with the DNA nucleotides. So it would take it out and replace it, and it just goes through the whole strand and does this until it's completely finished, and then also on the leading strand, it does that. And on the leading strand, it does the same thing. It removes the primer and replaces it with DNA. So it would go through and replace it just like it did for the lagging strand. And then next, we have ligase. So next, we have ligase, and that comes through, and wherever there were little nicks left where the RNA was replaced with the DNA, it creates a sugar phosphate bond so that they are connected together. So it does that and makes sure that everything's connected.